Let's review triglycerides before we begin looking at lipid metabolism. Recall, triglycerides consist of a glycerol molecule bonded to three fatty acids. Lipid metabolism involves lipolysis, the hydrolysis or breakdown of triglycerides into glycerol and three fatty acids. Beta oxidation, which occurs in the mitochondria. This is the breakdown of the fatty acids, 2 carbons at a time, forming acetyl-CoA. Acetyl-CoA, recall, transfers the 2-carbon acetyl group to the citric acid cycle. Ketogenesis. When elevated levels of acetyl-CoA occur, the extra acetyl-CoA can be used to produce ketones, a form of ketoacids. Elevated levels of ketones can lead to ketoacidosis. Lipogenesis. The formation of triglycerides from carbohydrates and proteins. Triglycerides are stored in your adipose tissue. The hormone glucagon triggers the adipocytes to break down the triglycerides into glycerol and fatty acids through the process of lipolysis. Let's follow the blue arrow. Glycerol can be converted into glyceraldehyde 3-phosphate, an intermediate of glycolysis. Notice that the arrow from glyceraldehyde can go up or down. In the liver, the glyceraldehyde 3-phosphate can be converted back into glucose to help maintain blood glucose levels. In addition, glyceraldehyde 3-phosphate can be converted into pyruvic acid as it continues down glycolysis. The fatty acids are broken down 2 carbons at a time through beta oxidation to produce acetyl-CoA. The acetyl-CoA transfers the acetyl group to the citric acid cycle, and ATP is synthesized to substrate-level phosphorylation, as well as carbon dioxide is produced. NAD and FAD are reduced into NADH and FADH. The NADH and the FADH go to the electron transport chain, and the high energy in the electrons that they are transporting is used to make additional ATP through oxidative phosphorylation. In addition, the acetyl-CoA can be used to synthesize cholesterol when the body is in need of steroids or bile salts. Elevated levels of acetyl-CoA will stimulate ketogenesis, producing ketone bodies. In addition, when proteins are broken down into their building blocks, the amino acids, some of those amino acids can be converted into pyruvic acid, which can then go back up glycolysis to synthesize glyceraldehyde 3-phosphate, which can then be used to make glycerol, or it can continue up to make glucose. In addition, some amino acids can be used to form acetyl-CoA, which can be converted into ketone bodies as an alternative energy source or enter into the citric acid cycle. So you can use amino acids to make triglycerides, and you can also use carbohydrates to make triglycerides. Let's review. What happens if you have too much acetyl-CoA or a diet that has no carbs? Remember, elevated levels of acetyl-CoA, which can result in a no-carb diet because the citric acid cycle shuts down, will stimulate ketogenesis and the production of ketone bodies. This can lead to ketoacidosis if the levels of ketone bodies are elevated. This occurs during uncontrolled diabetes mellitus starvation, and a no-carb diet.